without further ado, I want to introduce Sarah. First of all, I came across slime molds myself this last year when I was hiking and became really interested about this fascinating creature. So when I saw that Sarah did a blog on slime molds, I asked her whether she would be able to talk about this and speak to this. And little did I know that she worked with slime molds at Dr. O'Neill's Ray Collins lab after she graduated from college at UC Berkeley. And this is in the late 1970s. So slime molds were very interesting to a lot of people at that time. She has since earned master's degrees in botany and creative writing. And for the first half of her career, she managed the University of Washington Herbarium. And for the second half, she worked on biodiversity conservation and salmon recovery in state government. She served as the president of Washington Native Plant Society in the mid-90s. And currently she participates in several Native Plant Society committees and has curated the Botanical Rambles blog for Washington Native Plant Society since the blog began in 2012. So phew, that's a lot. So Sarah, take it away.
Okay, well, thank you. Thank you everybody for being here tonight. As mentioned, my name is Sarah Gage, and I'm speaking to you from Seattle, Washington. And I would like to acknowledge that my house sits on unceded ancestral lands of the Duwamish people. And they were the fierce people of Seattle. And I honor with gratitude the land itself and the Duwamish people, past and present. So before we get started with my slides, we're gonna have a little poll, because I'd love to know a little bit more about all of you who are out there in internet land. And so it's about 6 questions, and you'll need to scroll down to complete the poll. Of course, you don't have to answer any particular question. You don't have to participate in the poll at all, but I'd love to know a little bit more about you. So let's go with the poll if we can, Denise.
We forgot the Jeopardy music.
So please go ahead and select your answer. As you can see, there's a scroll bar here to get all the way through. Exciting, we have some folks outside of Washington. Wow. As you can see, all of the questions are being answered. And we're going to watch as more of you vote before I end the poll.
So I'm seeing some messages, Denise, that it's sounds like it's not letting people submit.
Just go ahead, and if you registered to attend, you should be able to submit.
Looks like maybe they are wanting you to answer all the choices. You can only submit if you have to, oh, so you do have to answer all the questions. I'm sorry.
We're up to 193, 195, okay. 200. So we're getting close. We'll just let it go for a few more seconds. I think we have a good sample of this giant group. So thank you all for being here tonight. And I'll go ahead and end the poll.
And I want to thank you for doing the poll, even though I guess it's been a little frustrating for some of you, but let's see. So looks like, how much do you know about slime molds? The majority is coming in. About half of you are saying a little bit. Okay, and we don't have any experts on, or at least confessing to be experts, or who could maybe submit their poll. So anyway, all right. And then let's see, have you ever seen a slime mold in the wild? Looks like most of you have, 70%. Okay, and some of you aren't sure. And then have you ever seen a slime mold in a class or lab setting? And that looks like most of you are saying no. And then have you watched any of the following? The recent NOVA program, Secret Mind of Slime, The Creeping Garden, which is a British documentary, or another TV program. And maybe some of you had to submit that because, or say that because it wouldn't let you submit if you didn't check something, so. And then, are you a member of the Native Plant Society? It looks like 30% of you are, and 63% of you. You know, I was joking as we got started that we may have to start the Washington Slime Mold Society. And then where are you watching from? Most of you are here in looks like 40% in the Puget Sound area. About 37% are from other Washington State locales. And I've seen people from Iceland and Tasmania. So welcome everyone. I'm going to close that and I'm going to try to disappear here a little bit. Let's see. I'm going to show you this. There we go. And I think we were having some bandwidth issues, so I may disappear after a while. But let's see. Here we go. Stop video. There we go. All right. So whatever your reason is for being here tonight, you're in the right place. We'll have a little chat about slime molds. And I hope to have time to take your questions at the end. And feel free to type your questions in the chat. If you, the chat's a little easier for Elizabeth, who's going to help me later on with the questions. So you can certainly do Q&A, but the chat box is easier. So, but I plan to plow through my talk and take questions at the end. Now, if you click on the raise hand, let's see, that's not an option, so forget it. I won't even mention it again. So let's go along here. Oh, let's see here. There we go. So I have here to report a stunning coincidence. This is me in Dr. Collins's lab at the Zeiss Universal Microscope with scanning cytophotometric unit. I'm the one in the plaid shirt. And this picture was taken on October 6th, 1979. So exactly 41 years ago. So that's kind of trippy that that was exactly 41 years ago. So here I am in the slime mold lab, and I'm measuring stained slime molds for the amount of DNA that they contain. And this was before molecular techniques became widely used. And so this was kind of state of the art at the time. And as I mentioned, I was in Dr. Collins's lab, and this is a picture of him from his obituary in Mycologia. When I knew him, he looked very much the same, except that he had big hair at the time I knew him. And it was my first job out of college with my freshly minted botany degree. And he was a great guy. He was a tall, jovial man, and he taught the mycology course that I had as an undergraduate. And I learned a lot from him. I probably could have learned more, but as you saw from my previous picture, I was in my 20s and I was busy with the other developmental tasks of an early 20s-year-old. And so, you know, something like in the Dylan song, music in the cafes at night and revolution in the air. So I will confess that I haven't spent much of the last 41 years looking at slime molds, but I've always had a soft spot in my heart about them. And I've always loved dropping them into conversation. It's always gets people get pretty interested when you say your first job was in a slime mold lab. So the inevitable question, of course, is what is a slime mold? And if you read the introduction to this talk, they're not plants, animals, or fungi. And if you're not quite sure what delimits those groups, that's also an interesting study. But so they do resemble fungi in some respects. And so they're typically studied by mycologists. And in the past, of course, mycologists were housed in departments of botany. Sadly, at least here in the United States, at least on the West Coast anyway, that I'm most familiar with, most departments of botany have now been subsumed, like as you'll see, slime molds subsume things. They've been subsumed into biology departments, but that's a different topic. So they're typically called myxomycetes, myxa meaning slime, and mycetes meaning fungi. They've also been called mycetozoa, which means slime or fungus animal. And like, as I said, I learned about them in my mycology course at UC Berkeley when I was an undergraduate. And however, now of course they're classified with a group that's known as the amoebazoans, also known as protists, or at least it's a group within the protists. And so they're an early branch of the tree of life, and they have true nuclei. So they're U, meaning true, karyotic nuclei. And what does that mean? Well, a true nucleus is a membrane-bound structure within a cell that contains the chromosomes. And that contrasts with prokaryotic organisms, for example, bacteria, that lack that envelope-enclosed nucleus. And the nuclei will be important a little later on as we talk about the life cycle of the slime molds. And then I also just wanted to point out that tonight we're talking about the myxomycetes, which are the true slime molds, which are also known as plasmodial slime molds. We'll talk about plasmodia in a minute. Acellular slime molds, we'll talk about that in a minute. And another name that you might hear referring to this group is myxogastrids. But so we're not talking about cellular slime molds. And that is probably most familiar to most people as an organism called Dictyostelium. And those cellular slime molds spend most of their lives as a separate single-celled amoeboid protist, so amoebae. And when they release a chemical signal, the individual cells then aggregate into a great swarm. That swarm then sort of sporulates, and because of that signaling and aggregation, they're of great interest to cellular and developmental biologists. And it's a good lab system for understanding how cells interact and to generate a multicellular organism. But so we're not talking about Dictyostelium and the cellular slime molds tonight. We're also not talking about slime nets. This is a group of which Labyrinthula is the one that you probably might have heard of if you've heard anything about this group. And they cause a wasting disease in eelgrass or Zostera marina. And they're not technically considered to be slime molds, but they have been thought to be slime molds in the past. So that's why I mentioned them. But in any case, we're not going to talk about them anymore. So myxomycetes, they have fascinated people for a long time. And the first scientific reference was in 1654. Ernst Haeckel was an artist and a naturalist, and he included this plate in his Art Forms of Nature in 1904. And these are stylized images of slime mold fruiting bodies. And so it's just I think the poster of this was posted in every mycology classroom that I ever sat in on. The other thing I wanted to say about this is that there's about 1,000 species that have been described worldwide of the slime molds. And they've been traditionally divvied up into about 6 groups, 6 orders, but recent work has shown that this is going to need to be revised. I'm really not going to emphasize that kind of classification tonight. We're going to just be looking at some basic shapes and life forms. So this is the life cycle diagram of the myxomycetes. And we'll start over here on the right. You'll see in this asexual part, the spores germinate and give rise to these what are called amoeboflagellates. So they can have a life as an amoeba, which means they just kind of ooze around as a single cell, or they might extrude they have 2 flagellae that help them swim. They're not really powerful swimmers, as I recall, and they swim in kind of a corkscrew motion. And most of us will never see this unless we're in a laboratory setting because they're tiny. So one of the things we were studying in Dr. Collins' lab was the various mating systems that the slime molds had. And so you'll see these guys have the red nuclei, and over here comes somebody with a blue nucleus. And so those then can fuse. And so the cells fuse, that's the plasmogamy, and the nuclei fuse, that's the karyogamy for K, and it forms the diploid amoebozygote, which are kind of red with blue specks or kind of purple. So then these divide. And so this is where this acellular comes from, because they are single-celled in that the nuclei divide, but it's just all one bag of nuclei. In our cells, when the nucleus divides, then the cell itself divides. And so you get two cells, more like up here, two single cells. But here, these nuclei divide and divide and divide. And so you have a multinucleate plasmodium. And don't worry, we're going to see a lot more about plasmodium. The other thing that's kind of trippy about this is that the nuclei all divide simultaneously. So some massive amount of signaling is going on in there. So we're going to look at this part of the life cycle now. So hang on, we'll get back to the other part in a second. So this is the Plasmodium of Physarum polycephalum. If you saw the NOVA special or the NOVA program, they pronounced it Physarum, which perhaps is correct, but I learned it as Physarum, so that's what I'm going to say. And polycephalum means many-headed. So you can imagine, you know, this is the leading edge of the Plasmodium. All these bumps are reminding people of heads. And this is what the researchers on that NOVA program are using as their lab rat. It's a relatively easy to grow in the lab, and so it's a very commonly studied organism. One thing I'm sorry I can't show you tonight is the protoplasmic streaming. I had hoped to show you a video tonight, but my version of PowerPoint doesn't support videos anymore. But please do look it up online. There's lots of YouTube videos of cytoplasmic streaming in the slime molds. And the real ones are, it takes about a minute. So it's all gushes one way, and then it gushes back the other way. And that takes about 60 seconds for it to gush one way and then reverse. And you'll find a lot of those where it's speeded up. And you'll notice if you did see the NOVA program, all of that was very much speeded up with time-lapse photography. So this is what you might see if you saw a Physarum polycephalum plasmodium in the wild. It's on a log and you see these veins. It's going after its prey or food. It tends to engulf bacteria, mold, spores, kind of whatever's small and tasty. I did read, so that you find them most often, just in general as a group, you find them most often on dead wood, leaf litter, or emerging from under bark. And sometimes you'll see them on bark chips particularly. Like if you have a garden where you put down wood chips, sometimes you'll see plasmodia on those. And one plasmodium down in Texas was documented about being 3 feet in diameter. This is what you'd see if you had a voice chamber that you brought in. So here's a piece of wood and they put in a piece of moist filter paper. This is in a Petri dish. I had hoped that some of the slimariums that I had set up would have something going on for us tonight, but they don't. I'm just generating some mold. But anyway, so in the lab, what you feed your slime molds with is usually sterile pieces of oatmeal. That's certainly what we did in Dr. Collins's lab. Now, this is probably what you most likely have seen in Washington State. This is probably not Physarum, but probably the notorious dog vomit slime mold. And we'll see more of this guy in a little bit. I want to talk now about the second half of the life cycle. So after the plasmodium oozes around and engulfs bacteria, and that's one of the reasons why these are different from fungi. Fungi absorb nutrients instead of actually engulfing them. So they're engulfing these whole bacteria through the process known as phagocytosis. But so then when things say get a little sketchy in the food department, or starts drying up, or you know, conditions change, the season changes, some trigger is reached, some developmental biology trigger is reached, and the slime mold will fruit. Now, fruit is not the technical term, because you know, a fruit is a swollen ovary of a flowering plant that has seeds inside. But these fruiting structures have a stalk or a stipe, and then inside, this is where meiosis takes place and the spores are produced. So it's that reduction division, so it gets it back to the haploid state. And inside, and we'll see plenty of pictures of this later, there's capolitium, which is these kind of hairy things that are thought perhaps to help the spores disperse in air. And this is an example of a particular genus, Stemonites, which has these sort of elongated fruiting bodies, and we'll see other forms later on. So here are some slides. So this is an example of photographs of, so Stemonitis. So it's been in its plasmodium and now it's kind of gathering its resources. It's gushing all up and it's going to start to fruit. So here you see it's kind of gathering itself up into these long pipe-like kind of tube-like structures. It's still pretty gushy here. And then this is what you might see more typically in a dried out stage. So it's got these very hair-like stalks or stipes and these chocolate-colored tubes, and that's where the spores are. And this can actually, if you were to flick it, say with your finger, if you came across it on a dead log, the spores would kind of poof out, kind of like a puffball, if you're familiar with those types of true fungi. Afterwards, you get something that looks like this. This is a beat up one. I'm proud, I actually took this picture. I took it at Carkeek Park just after I had agreed to give this talk. And I started looking for slime molds, and what do you know? I found this one on a log at Carkeek Park. So this is a kind of beat up Stemonitis. And you can see, if you look closely, you can see the columella, the stalk that's within the sporangium. And then this kind of fuzzy stuff is the capolitia, the hairs within the sporangium. And if you were to look at the capolitium under a microscope, this is what you would see, those hairs. And then the little spherical things are spores still captured within that capolitium. So after the release of spores, sometimes you can see these internal structures. Another species is this Arceria. And here you can see the capolitium is just sort of oozing, popping out of the top of the sporangium, the sporophore there. And inside there, you can see in this case, the capolitial hairs are kind of ornamented, kind of have spiral structures going around them. And here's the spores. And the spores are tiny. They're about 7 to 12 microns, which is less than half the diameter of a human hair. So pretty small. They can be variously ornamented, not as maybe strikingly as, say, diatoms or pollen, but it is an aid for identification. And in some cases, you need to see the spores in order to identify species or the microscopic features of some of these other structures. And this slide is to remind me to tell you that slime molds aren't alone out there. They are preyed upon by certain insects, and then the insects also play a role in dispersing the spores. Spores are also distributed by air and by water. And many of the species are cosmopolitan because these spores are so tiny, they can get up into the airstream and go pretty much everywhere. And so wherever conditions are suitable, you're likely to find certainly some cosmopolitan species of slime molds. I want to tell you a little bit about one of our forebears in the study of slime molds. This is Julielma Lister, and she wrote one of the early monographs on slime molds with her dad, Arthur Lister, and then after he passed away, she kept up with the revisions. She was also an artist and illustrated the volumes. And I'm going to read you a few things about her field techniques because she has such a nice way of phrasing them. So, localities favorable to Mycetozoa. Mixed woods on not too steep a slope should form good hunting grounds for Mycetozoa at any time of the year. Here, old logs and the underside of fallen branches should be searched for many species of Physarum, for the brown feathery tufts of Stemonitis, the dark pin-like sporangia of Comatrica, the clustered yellow balls of Trichia, or the crimson buff and gray clubs of Arceria. In heaps of dry leaves or dead leaves, sporangia are to be found below the outer layer and not deep down in the heap, since the Plasmodium come up to near the surface to fructify, meaning to fruit, in order that the spores may have a chance of being distributed by currents of air. She goes on to say, it should be remembered that the sporangia of Mycetozoa are, for the most part, minute objects, not as much as a millimeter in diameter, and need careful looking for. They are so fragile that a shower of rain may wash them away, leaving hardly a trace behind. But on the other hand, on any day in a moist weather, fresh Plasmodium may emerge and provide a new supply of fruit. And she has some words of wisdom for us about collecting. She says, on setting forth to collect Mycetozoa, it is well to be provided with a strong knife and a basket or tin in which smaller boxes may be packed. Some of these should be lined with cork, that the slip of wood or leaf bearing a fragile specimen may be at once pinned down firmly, and the sporangium walls preserved from injury. Immature sporangia should be brought home with as little shaking as possible in a tin box and then kept moist, not wet, until they are quite ripe. Some ventilation should be allowed, and they may be attacked by mold. When found in the plasmodial stage, Mycetozoa are often difficult to deal with, as they are so sensitive that in spite of all care, they may not recover the shock of being moved from their natural surroundings. And finally, she says, once the sporangia of Mycetozoa are mature, they should be kept dry. Many valuable specimens are ruined by mold from being kept shut up in a damp state. They can be perfectly preserved as dry objects for 100 years or more if they are guarded from two enemies, damp and insects. So I thought we'd take a look now at some myxomycetes, some slime molds that we might see here in Washington State. So here, for example, is Arceria denudata. And these identifications are all from the people who took the pictures and who kindly put their photos onto Wikimedia, which is where I got the bulk of the photographs that you're seeing tonight. So I'm not making any claims that the identifications are correct or incorrect. And I just put them forward as this is what the photographer said that they were. So this one has the common name of carnival candy slime mold. You can sort of see that the pink capillacea there look kind of like cotton candy, as we would call it. It also kind of reminds me of Marge Simpson, her hairdo on The Simpsons. But it starts out pinkish red, and then it can age to a browner color. Another one that I think is just adorable is this Craterium minutum, these little marching across the leaf. As I mentioned before, unfortunately, most species require microscope works because of the spores or internal structures, but you can probably get them to genus by eyeball. Another member of this genus is Craterium leucocephalum, and this has this wonderful, so this outer case of the sporangium is called the peridium, and this has a kind of a little door that opens up called an operculum. And so it has that sort of door that opens up to let out the spores. This one grows on dead leaves and twigs, which, you know, a lot of them do, most of them do. Another one I have always loved is this Cribaria in this case. And this has, so all of the spores are gone, and what's left behind is this wonderful network what they call a peridial net, which is like little cages or lanterns. And I've just always thought that that was so beautiful. Many of the slime molds, especially in the Fissurales, that is the genus that contains Fissurum, or Didymium, which is the group that Dr. Collins and I worked on in his lab. We worked on a species called Didymium iridis. Anyway, they have this so-called lime incrustations. It turns out that it's actually calcium carbonate, not, as I understand it, calcium oxide, which is the true lime. But anyway, in some of them, they will have crystals. Some of them will have more like granules. This lime characteristic is something that you'll see frequently in keys to the slime molds. You'll see also that this one has a much shorter stalk or stipe, so much so that in some cases, I don't know about in this species, but in some cases, they will be what's called sessile. That is, they will have absolutely no stalk. They'll just be like a little blob on the substrate. This one has another puffy kind of capillicium. And I mentioned that Dr. Collins had big hair when I knew him at Berkeley. He had an afro, and he was in class. I remember him patting his afro and saying that it was similar to a emerging capillicium, that it was getting bigger with maturity. And this is Laocarpus fragilis. Even the photographer said maybe on this one. It's very colorful. It's not quite mature in this picture. And this one apparently can look quite a bit like insect eggs. And this one, they do say, is more likely to be found in coniferous forests rather than deciduous forests. So perhaps a good candidate to find around here. So these have all been sort of your typical, they have a stalk and a sporangium on top, a sporophore of some kind. Now, another type of fruiting body is seen in these. This is what's known as an athelium. And in this case, it's a sessile structure, that is, it has no stalk, but it's also quite large. It's much larger than a little sporangium. And so it's kind of the plasmodium just hunkers down and fruits in place. So that's called an athelium. And in this case, it's quite mound shaped. And these start out as pink or salmon colored. And this one's called, its common name is the wolf's milk slime mold. And I couldn't find any information, maybe you know why it's called wolf milk. But I also did find a lot of pictures of people poking this and getting out some pink ooze. So I'm pretty sure that wolves don't have pink milk, but perhaps that has something to do with where the common name came from. These can become quite orange red, and this is one that I have seen locally, and in the ones that I have seen, it has had this very reddish orange color to it. And then they can mature to kind of this dark gray or almost blackish color. And this is when it would be sort of drying out and the spores would be maturing. Now, one I have never seen in the wild is this sort of pretzel-like one. It's called the pretzel slime mold, Hemitrichia serpula. And in this case, the fruiting body is known as a plasmodiocarp. Again, where the plasmodium just kind of sets down where it is. And you can imagine the veins and so forth from the plasmodia just sitting down and sporulating in place. And this has, the loops on this are about 4 to 6 millimeters in diameter, so about ½ an inch in diameter. This is the so-called coral slime mold, and this has kind of a completely different kind of fruiting body, and is now considered perhaps a sister group to the true slime molds. I don't have a good micrograph of this, but if you can sort of see how those little white fingers are kind of fuzzy, and so on each of those fingers, then spores are being born externally and individually. So it's almost like, so they don't have a covering over them. They're sort of just sticking out there, and it's quite a different shape from the other slime molds we've been looking at. And these can get to be pretty good sized colonies. You you can see, I think from this picture more why they might be called the coral slime mold. So now let's talk about some big ones. And these can be quite alarming. As I mentioned, there was that 3-foot across slime mold in Texas. I don't believe that the slime molds were inspirations for the initial Blob movie in 1958 or the remake in the 60s. And I'm pretty sure that they weren't the inspiration for the slime people, but you just never know. So I think there's ample opportunity here for some additional films in the canon of slimy and blobby films. So let's get cracking with those screenplays. So this puppy's called Mucilago crustacea, and this is a bigger type of ithelium where the Plasmodium just kind of hunkers down and starts to fruit. And this has crystals of those limey or calcareous crystals in its structure. Sometimes a Mucilago can be found growing up the stalks of grass. And I noticed that the WSU extension has a section on what to do if you've got slime mold on your grass. And their suggestion is just to A, leave it alone, or B, if it really bothers you, you can just hose it off. And you know, the slime mold will just go off, creep off somewhere and regather its forces and live to sporulate another day. Mucilago also will just crawl right up on plants and form these kind of things. They remind me a little bit of cattails when they've gone to fruit. But so this would be kind of a crusty fruiting body here, quite large, as you can see from the leaves of the plant. And this is probably the most famous of them all, Fuligo septica, the dog vomit slime mold. Again, it's find it on decaying logs and wood chips, mulch, as I mentioned. It's also sometimes called the scrambled egg slime mold. And here you can see it looking more like scrambled eggs than like dog puke. They can be yellow or sort of cream colored. Sometimes as they start to fruit, they get kind of this tannish. And the spores themselves, which you can see breaking through here in that mature crust, are blackish. Sometimes the ithelium can be sort of orangish or kind of brown tannish, and they can look sort of like animal droppings. And then, of course, they might just be coming for your beer. Well, I've been doing a fair amount of talking. And so now one of the things that I have learned from the Facebook group, which I'll give you the link to later on slime molds, is that a lot of the traffic on that Facebook page is this a slime mold that I've taken a picture of or not? And so now I'm going to say, have you take a quiz. Now, this isn't the poll. This isn't a poll. So I need you to take a piece of paper. And there are, let's see, I think I put, yep, I've got an even dozen of pictures here for you to take. So take out a piece of paper and write your numbers down 1 through 12 and keep track of your answers. Now, of course, in person, if you were to look at these and not just on a photograph, you'd have a better sense of what the substrate was and whether, you know, it was in even a slime moldy kind of place. So let's just see how you do. And I'll give you just a few minutes on either. So here's number 1, slime mold or not. And here's number 2. Yeah, I do need some like game show music. Deet, 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 deet. Here's number 3. Number 5. Number 6. Number 7. Number 8. Number 9. Number 10. And 11. And for our last one, number 12. All right, let's see how you did. So number 1 was our pal, the dog vomit slime mold, Fuligoseptica. Number 2 was some stink bug eggs I saw on a columbine in my garden this summer. Number 3 was a Plasmodium. Number 4 was the wolf's nut slime mold, Lycogala epidendrum. Number 5 was some paint I saw on the ground on my walk yesterday. Number 6 was a German chocolate cupcake. Oh, how I wish I had one now. Number 7 was the carnival candy slime mold, Arceria denudata. Number 8 was dried tree sap. Number 9 was lichen on tree bark. Number 10 was chocolate tube slime mold, Stemonitis. Now 11 is actually one that I submitted to the slime mold group on Facebook. And it turns out that it's a member of the ascomycete group of fungi called brittle cinder fungus. And you can see it was kind of black. That's Kretschmeria doista. And then the last one was coral slime mold, Serratiomyxa fistulosa. Well, if this has just whet your appetite for more about slime molds, I wanted to give you a few resources. Probably the most available book on slime molds is a good one. It's Myxomycetes, a handbook of slime molds. I was able to get it from the public library. It's also readily available online. And it has keys and descriptions, as well as information about the various structures of slime molds, collecting, their distribution and ecology, and it's fully illustrated. The book on the right, a companion to cosmopolitan slime molds, is co-authored by one of the same authors, Stephen Stevenson, and an artist, Angela Maley, who's done these wonderful illustrations, very beautiful illustrations. And this is a self-published book that she's done, but if you look it up online, you'll be able to find her website, and her drawings and paintings are very lovely. If you're interested in more of a narrative experience with slime molds, I highly recommend Kelly Brenner's Nature Obscura. She's got a chapter on slime molds. And this was kind of the impetus for my writing my blog post on slime molds. I highly recommend Kelly's book. Again, it's a narrative discovery, as she said, of a city's hidden natural world. It takes place in Seattle, so it's very relevant to those of us who live in Western Washington. And then if you've got some kids in the family, or if you just like to start out with the basics, this is another book I got out of the library, Creeping Slime Molds, and it covers all the bases. So it's got some good pictures and just the right amount of explanation. The online groups that I was mentioning, there's a Facebook group called Slime Mold Identification and Appreciation. Instagram, I just found this out, has a hashtag Slime Mold Sunday. Those photographs, people there are really, really beautiful. And one of the things I really like about the Facebook group is the patience of the people who obviously been at this for a while. So a lot of people, I think, signed on after they watched the Nova special the other night, and are posting a lot of things that people are patiently explaining to them. Nope, not a slime mold. But so that's a great place to get some of your questions answered. And then I also came across the site mushroomobserver.org. There's a number of people there who are slime mold aficionados and post beautiful photographs. And then iNaturalist.org is a community science effort documenting and identifying all types of organisms around the world. And they have a hefty slime mold presence and representation on that. All of these are global in scope. And so you can find slime mold aficionados all over the world, which is where slime molds grow. So with that, I'm going to go to my last slide, and I think we have time to take a few questions. It's about 10 of 8. And I've also put my Native Plant Society email address on here. If you don't get your question answered tonight, or if you have something you want to address directly to me, feel free to email me at that address, and I'll do my best to either answer your question or help you figure out where you can get an answer.
Okay, I am back with some questions. Excellent talk, and people in the chat, I don't know if you can look in the chat box, but people enjoyed the quiz. We had comments like, woohoo, and what do I win? And stuff like that. So I've been taking down some questions. There were a couple people that wanted to know if they are edible by humans.
I have read some reports of some edibility. I'm pretty sure they're not toxic. And as I recall, there was a book that I remember back from the 70s that talked about growing them in liquid culture, sort of plasmodial globules, which I have to say I didn't investigate too much because I thought it sounded really not like something I would want to eat. But yes, I think there have been records of people eating them. I don't think they're a big part of any particular cuisine. And I don't know much about that, but yes, I do think that at least some of them are edible.
Thank you. Another question was, it's a sort of interaction with the rest of the world. Do they have predators?
My impression is, yes, they do. I'm trying to think. I think certainly some slugs and snails will eat them. I think it's incidental. I don't think that there's any other organism that makes its living entirely by eating slime molds. But I think that in that great sort of mush of decaying and, decomposing and digestion, that there's a lot of interactions. I don't know more about it than that, but yeah, they're definitely part of sort of that whole interaction there. And as I recall, and I could be wrong on this, I'm not remembering that there's anything, like I say, that makes its living only by eating slime molds.
Okay, thank you. A couple of people wanted to know if they are used in any human way, like similar to how fungi are sometimes used in pollution control, or if they have any other sort of domesticated uses.
Not that I'm aware of. They are hugely important in certain types of laboratories. So and the NOVA special goes into that quite a bit. So they have a utility in terms of scientific experimentation and understanding about movement. One of the things I didn't mention is that the Plasmodia actually have the same molecules that mussels have. So actin and myosin are involved in that cytoplasmic streaming and how the Plasmodia actually move from place to place. So they're an interesting model system from that respect, as well as they seem to have a sort of, they call it kind of an intelligence in that they can solve like maze problems. If they perceive a food source, they will find the most direct way to get there. But as far as like remediation, pollution remediation, or anything like that. I'm not recalling anything in my reading about that.
Okay. Somebody wanted to know if they have an aquatic phase. I think prompted by the fact that they're often, that they can get washed away if they get rained on.
Mm-hmm. Yeah, I think that they're preferentially terrestrial. I believe there are some aquatic and perhaps even some marine, some organisms that can tolerate or even thrive in a marine or aquatic environment. I don't believe that they're ones that we have commonly here in the Northwest. I think that was something that I read maybe sort of Australia, Tasmania, New Zealand. I'm kind of at the limits of my knowledge about that.
Okay. Here's a couple of related questions about identifying them. Are any slime molds fluorescent? And how do you tell a slime mold from a fungi, from fungi?
Yeah, I'm not aware of any slime molds that are fluorescent. I haven't come across that in any of the reading that I've done. I've certainly never seen a fluorescent slime mold. I think that would be pretty, especially in the plasmodial phase, that would be pretty exciting. You know, something glowing coming at you at night. And the other, oh, how do you tell them from a fungi? Okay, so there's two major groups of fungi that we see most frequently: the basidiomycetes, which are like mushrooms, and the ascomycetes, which are like yeasts and other types of molds, like some of the cup fungi. And then there are things like molds like you might find in your refrigerator, which are usually asexual forms, mostly, I believe, of ascomycetes, but sometimes of basidiomycetes. Or sometimes they're just completely asexual. We don't really know where they belong. But what the true fungi have that slime molds don't have is what they call hyphae, which are threads of fungal cells that creep along. If you have ever seen like how an orange will get moldy, those are the hyphae that are spreading over the surface of the orange. So I think one of the ways to tell would be, so for example, if you've got something that's kind of thready looking, like most of the slime molds, at least until they pop their tops and spread out their caplitia, they're not going to be very thready looking. And the plasmodia aren't very thready looking. They're more globby and kind of, you know, slimy. Whereas, and then, so the other thing with fungi is that if you have anything that's like a true cup, like lichens sometimes have cups, and theirs of course are a symbiosis between an ascomycete usually and usually a blue-green alga. So a cup or like a fungus, like a mushroom, or a conch, like sticking out of a tree. So so that's how I tell. So I think it's a matter of just kind of honing your observation skills, getting comfortable with what does a fungus look like.
Okay. We're just about at 8 o'clock, and so I'm going to stop with a few housekeeping questions. Somebody asked if we could repost the names of those movies, and I think the best way to find those back would be to watch the video, which, so this evening has been recorded, and the video will go up on our website within about 48 hours. And if you have additional questions, please go to Sarah's blog, Botanical Rambles at WNPS.org. And I'm just looking to see if there's any other last minute questions. I think this is from somebody maybe on the Facebook group. She says, we encourage people who post slime molds to return over a few days to see their progress. Somebody's saying they've seen.
I know what that's about, Elizabeth.
Okay.
So if you do run into a plasmodium or something, you think might be a Plasmodium. On the Facebook page, they really encourage you to go back and take pictures at subsequent times. So later in the day, the next day after that, because having those different developmental stages documented is very useful, both for identification and then also just for general knowledge sharing. So yeah, there was definitely a call for that with the people who have been administering the Facebook page. So yeah, if you see something that looks interesting, yeah, go back and take more pictures, because that can be important for a number of reasons.
Okay, here's a quick question. My classes use Physarum a lot in high school classes. Are there any known problems with students coming in contact with the Plasmodium or fruiting bodies, example, infectious or allergy issues?
I have never heard of any problems with that. You know, people can be allergic to anything, I suppose, but yeah, not that I'm aware of.
Okay, and then I'm going to finish with, oh, here's somebody who went back and checked out the movies. So get your pencil out. One of them is called The Secret Mind of Slime, and the other one is called The Creeping Garden. And then I saw a comment somewhere that I would like to finish with, if I can find it again. Anyway, somebody said they had seen a fluorescent slime mold.
Oh, that would be exciting.
Oh wait, here it is. I have seen a fluorescent orange plasmodia that developed into tiny tangerine-like sporangia, but I don't know if there are any bioluminescent slime molds. So on that bright and intriguing note, I'm going to call it quit to the questions, and thank you very much, Sarah.
Yeah, you're welcome. Always good to end with an unanswered question. So let's go out there and find out more.
Great.